Hello and welcome to Prac 1 for Cell Biology. In this practical class we'll be doing two different experiments. The first one will be looking at setting up a serial dilution of common yeast cells, and in the second half of the prac we will be taking a swab of the inside of our cheek in our mouth and looking at human epithelial cells, and that's quite fun because everyone will have an opportunity to look at their own cells. We should be able to see a nucleus, and we should also be able to see if you've brushed your teeth or not, depending on how many bacterial cells are present. So let's start with the serial dilution. So the first thing we're going to do is today we are using common yeast, which you would buy in the supermarket. We're looking at it, it's been grown up in a YPD broth, and in fact, of course, yeast is a unicellular fungus, a eukaryote. So when we look at yeast, if we took a drop of that yeast and put it on a microscope slide and had a look down the microscope eyepieces, we would see many cells, and it would be almost impossible to count the living cells. So we do this serial dilution setup in order to get an accurate measure or count of the living cells in the yeast culture. So let's get started. The first thing we will need today are 7 McCartney bottles containing the YPD broth, and of course we need our yeast solution, which we've grown up earlier. This is a concentrated yeast solution. Remember, it's not a problem today because this yeast is just the commercial yeast you would use to make bread or pizza dough. It is also a good idea not to wear gloves today because we are using the Bunsen burner today. And of course, if you combine the plastic of gloves with a flame, there's a possibility of burning your gloves to your hands. So today we wash our hands before the setup, and we'll wash our hands again after we finish the setup. So let's get started. The first thing we need to do is turn our Bunsen on, and you can see it's set to the safety flame at the moment. Let's open the Bunsen air holes. So we have the blue flame, the hot flame. Now today we're going to be using sterile technique, and obviously we don't have a cabinet. We're doing this microbiology on the bench. So the Bunsen burner is very useful. If we work in underneath the flame, of course heat rises, which means that when we take the lid off bottles containing sterile media or agar plates, it means that if we keep the lids off only in under the Bunsen burner, heat rises and hopefully air is pulling up things like fungal spores and bacterial cells, and they won't drop into our solutions or agar plates. So it's really important to only take the lids off the McCartney bottles and the agar plates in under this, what you would call cone of sterility. So let's get started. We have our yeast solution. We're going to take 1 ml of our yeast solution and add it to our YPD broth in each of these McCartney bottles. And these have been very nicely labelled from 1 to 7, 10 to the minus 1, up to 10 to the minus 7, as you can see. What we're doing is, as I said, a serial dilution. So we'll take 1 ml of the yeast solution and add it to tube 1, and this will be a 1 in 10 dilution or 10 to the minus 1 dilution. We'll mix this solution. Then we'll take 1 ml of the first dilution and add it to tube 2. And this will be a 10 to the minus 2 dilution or a 1 in 100 dilution. And so on up to 10 to the minus 7. So we'll take our pipetta, our Eppendorf pipetta, and it's set to 1000 microliters or 1 ml. So we need to pipette quite accurately for this to work properly. We put the pipette tip on the pipetta. Remember, we're only taking the lids off our vials or agar plates in under the flame. So we take the lid off our McCartney bottle. You can flame the edge of the bottle briefly, again to keep it hot. Remember from Fiona's hints when we did prac 4, we were pressing just to the first stopping point to expel the air. We lift our thumb slowly to take up 1 ml into our pipette tip there, and we'll take our first tube, which is bottle, which is the 10 to the minus 1 or the 1 in 10 dilution. Remember, we're adding 1 ml of our concentrated yeast solution to the 9 ml of YPD solution. We'll take the lid off this McCartney bottle, we'll flame it, we'll add our 1 ml, pressing the button all the way down with our thumb to expel all of the liquid from the pipette tip. We then flame the neck of the bottle again and screw the lid back on. So, we've created our 1 in 10 dilution. We can mix that, swirling it gently to mix, and then we'll take 1 ml of our 1 in 10 dilution and add it to tube 2, which will create a 1 in 100 dilution, or 10 to the minus 2 dilution. And we'll continue on, like I said earlier, up to the 10 to the minus 7 dilution. So I've just demonstrated one of those dilutions to you. You would do all of those dilutions yourself, maintaining sterility the entire way. We are then going to choose 3 of our dilutions to plate out onto agar plates. And whereas for the dilutions we used 1 ml in 9 ml of broth, now we're going to actually take a 0.1 ml or 100 microliter aliquot, and we have another pipette here set to 100 microliters or 0.1 of a ml. So let's take up 0.1 of a ml, and before I do that, let's talk about which tubes we're going to take the aliquot from. Of course, the most concentrated solution is the solution we started with, the concentrated yeast solution. But of course, the 1 in 10 dilution or 10 to the minus 1 dilution will be the next most concentrated, right up to the 10 to the minus 7 dilution, which will be the most dilute of the yeast cells. So we'll choose 3 bottles, and we usually would choose based on the TechStarts experiments, but today let's choose the 10 to the minus 3 tube, or the 1 in 1,000 dilution, the 10 to the minus 4, and the 10 to the minus 5. And we'll take 1.1 of a ml or 100 microliters from each of these McCartney bottles, and we're going to plate that onto these agar plates. And why are we doing this? Because we want to see how many cells grow, and we'll leave them to incubate at 37 degrees overnight. And tomorrow, or the next time we're together in the prac, we will count the cells, the colonies that have grown on the agar surface. So let's get started. Like I said earlier, we've set the pipetta to 0.1 of a ml or 100 microliters. Here's our 10 to the minus 3 or 1 in 1,000 dilution. Taking the lid off again, only in under the flame to maintain sterility. Flame the neck of the bottle. Press to the first stopping point, and you can see now that the pipette is set to 0.1 of a ml, that's much shorter distance. Lift your thumb to suck up 0.1 of a ml, and you can see the volume is much smaller. Flame the neck of the bottle and screw the lid on. So we have our Petri dish containing agar, and we're going to pipette this 100 microliters of yeast onto the surface of the agar. Pressing the button on the top of the pipette all the way. Again, I did that in under the flame to maintain sterility, and I took the lid off the agar plate for the minimum amount of time. What I didn't point out to you, of course, is that before we do the aliquoting, we should label our agar plates, and you should put the name of your group around the edge of the plate. So I'll put SC for my initials. I'll put 10 to the minus 4, which is the next dilution, and I'll put the date so that we keep track of when we did the experiment. And you can see that I put those around the edge of the agar plate, because when we come to count the cells, if we've written the label across the middle of the plate, it will be much more difficult to count the cells. So now what we need to do, of course, is spread those cells in the liquid around on the agar surface. And to do that, we'll use a glass spreader. So this is dipped in ethanol, and of course ethanol is very flammable, so we need to be careful with the Bunsen, keeping the beaker of ethanol not too close to the Bunsen that has an open flame. And just in case, of course we won't because we'll be very careful, but if you were to set light to the ethanol because you put the flaming spreader back into the beaker accidentally, we have this watch glass ready to put the flames out, but of course we'll be careful and we won't need that. So the glass spreader is dipped in the ethanol. We'll flame it briefly under the hot Bunsen to sterilise it. Of course now this is very, very hot, and if we touch the cells with the glass spreader straight away, it will kill the cells. So to cool the glass spreader, we again, taking the lid off the Petri dish only under the Bunsen, touch the edge of the agar away from the cells, and this cools the glass spreader so that it's safe. We then will spread the culture, the broth culture containing the yeast cells, thoroughly around the surface of the agar. Just to spread the cells to all the edges and so they're evenly spread on the agar surface. So now we have our 10 to the minus 3 dilution, or 1 in 1,000 dilution, and 0.1 of a mil of that was, or 100 microliters, was spread on the agar surface. What we need to do now, of course, is seal the plate using parafilm, and this is the stretchy kind of tape that comes in a roll. It's been cut very kindly for us by the tech staff. Peel the paper off and hold the plate in one hand and stretch the tape around. And as you can see, it tears very easily, but you can reuse it, and you can use that to seal the agar plate. So we now have our 10 to the minus 3 dilution spread on the agar plate, and we will place that upside down with the agar surface facing upwards. Why? Because of course condensation will form on the lids of the Petri dishes, and if we sit them facing with the agar on the bottom, condensation could drip down from the lid and colonies would all grow together. So we put the plates upside down and we will now incubate them at 37 degrees overnight for the yeast cells to grow. So we would do the same thing for the 10 to the minus 4 dilution, or the 1 in 1,000 dilution, and the 10 to the minus 5 or 1 in 10,000 dilution. We'd spread 0.1 of a mil or 100 microliters on each of those agar plates. We would seal them up with parafilm and add them to our stack here and incubate them at 37 degrees. Let's now look at some results that the tech staff have prepared earlier for us. Let's go to this light box. And in fact, we have an agar plate of dilutions from 1 in 10 or 10 to the minus 1. You can see the yeast cells have grown on the surface of that agar, right up to 10 to the minus 7. And you can see that there are no yeast cells growing on that agar surface because the yeast solution was so dilute. And in between, of course, you can see the 1 in 100 dilution. You can see how concentrated the yeast cells are. And you can see, for instance, the 1 in 10,000 dilution there, or 10 to the minus 4, and you can see colonies of yeast cells which are easily countable. Now, in your prac notes, if you read those before you came to the prac, you will have read that we can only count colonies that are greater than 30 or less than 300. And so we do these dilutions, and we've done 3 of them today, in the hope that one of our plates will have, or at least one of our plates, will have between 30 and 300 colonies on it when we come to count the cells. And you can see that probably this 1 in 10,000 or 10 to the minus 4 dilution has more than 30 and less than 300. And we would simply count those by using a marker pen and placing a dot over each colony as we count them. And you would get some sort of measure of the number of colonies growing on the plate. We'll then use a calculation with this colony count to calculate the number of yeast cells, living yeast cells in the colony. That will be provided for you. It's in the technical prac notes and your tutor will go through those with you.